Okay, so I guess we're live now, and we have a few participants in the room already. I'm gonna just wait for a couple of minutes for everybody to be here before we start. Or perhaps to save a bit of time, just going to start sharing a bit of the 2 minutes introduction. I'm going to be talking now in a couple of minutes. And really, I guess, thank you all for being here today. And, this is very exciting to be able to have, kick off a seminar series with 10x Genomics and Millennium Science, all things single cell technology and genomics. So while people is joining, I guess that we will have a few meetings. So watch the space where you got all this information and the advertisement for this seminar series. So watch that space, because we will be keeping that announcements and updating speakers as we go. But really, this is in the context of, as you all know, we have UTS new capacity that is being implemented. So earlier this year, we implemented the single cell technology facility, where we have a lot of equipment focused around single cell genomics. And this is really a joint venture between the Faculty of Engineering and Faculty of Science. And it's very exciting that it's coming together. Now, the lockdowns and everything has been a challenging time for everybody, including for us. But I'm very happy that this is sort of coming along, and we can get to do great single cell genomics in campus. So, so the intention of this seminar is basically a little bit of like an information session. But also to get a little bit of information to where you can go to speak to 10x Genomics about your design of experiments, if you're interested in doing single cell genomics. And the idea is to have a little bit of an overview on a specific technology. At the moment today, we're going to have a little bit of an overview of everything, but subsequent seminars will be more focused to specific technologies. And then we have another sort of invited speaker, which will be sharing their own views and their own research and their own expertise to us, showing what we can do with single cell genomics and the potential of that. So really ask questions. We have a chat. So if you have questions along the way, we're gonna have two presentations. I'm gonna present in a minute. We have Yvonne Peterman, which is the Science and Technology Advisor with 10x here in Australia. She's based in Melbourne. And we also have Luciano Martellotto from Harvard University, and he's the Scientific Director of the Single Cell Core over there. And both of them are very knowledgeable in this kind of technology. Ask questions. So we're gonna have the two talks, and then at the end we'll have a bit of a Q&A at the end of the second talk. But along the way, you can put the questions in the chat, and then we'll get to there at the end. I guess that we can start with, we have a few participants in here, so we can start with the presentation. So let me please introduce to Yvonne Peterman. So Yvonne finished the PhD in biology and genetics in the University of Freiburg in Germany. And she's been working in a number of different companies in life sciences. So she has a lot of expertise in different areas of biology, including specifically in genomics technology. And recently she joined the 10X team in Australia as a science and technology advisor. So she's going to tell us about the products and the technology on 10X and what we can do, what we can achieve. So Yvonne, over to you. Thank you very much for being here today.
Great, thanks David for the introduction. I'll just share my screen. Right, everyone, can you see this?
Yeah, all good.
Perfect, excellent. So yes, thanks David, thanks for the intro. Good morning, everyone. It's really exciting to be able to kick off today's meeting. And as David said, this is really designed to present to you the big picture of the capabilities and some of the new products that we also have in the pipeline. And then we'll dedicate the coming seminars to each of these assay types and more specific applications to actually really drill in. So I'm going to use the next 15 to 20 minutes to do this, and I just want to do and dive right in and talk about sort of our three complementary technology platforms, and because that's what we can sort of divide them into. And we begin with the Chromium single cell platform, and as you can see here, that consists of an instrument which is called our Chromium controller. It's quite small, like a shoebox, for some of you who haven't seen it. And what you do is you feed this instrument with this disposable microfluidic chip here that has 8 ports, so you can load up to 8 samples. And really in this instrument, you can actually perform these four main assay types, which we'll be covering in today's presentation. But what I want to focus your attention on is the sample input. So what we are doing is we are feeding this, or we are analyzing single cell suspensions or single cell nuclei suspensions. So they can really be derived from a wide variety of sample types and tissue types, from fresh and frozen. And there is a lot of demonstrated and tested protocols out there to utilize this sample type. But other than that, there's a lot of available protocols out there. And soon we will also have a product that's called fixed RNA for fixed RNA samples, which should make the accessibility of this technology even more exciting. The next of the platforms, or the second major platform that we have, is the Visium spatial transcriptomics platform. Here, unlike the Chromium single cell platform, there's actually no dedicated 10X instrument required. All you all you utilize is this glass slide here that you can see, where we put tissue sections on these 4 dedicated capture areas and analyze the transcript. So how this works is you place the tissue sections on, and the mRNA transcripts or the poly-A detected transcripts are going to diffuse down and are going to be detected on the glass slide. What you do need is an imaging system capable of taking H&E or immunofluorescence images, because this technology also allows you to co-detect protein at the same time. And this is available for fresh frozen as well as formalin fixed paraffin embedded tissues now. So quite a comprehensive solution that I am not going to go into any further detail today. So we are going to dedicate an entire seminar to this platform if you're interested in hearing more about it. And the third complementary platform is something that we are going to be, or that we are really excited about, which is our in situ platform coming up next year, enabled through our acquisition of Rico and Katana, where Also here we are using tissue sections, but instead of detecting mRNA on a glass slide, sequencing that and then mapping it back to the spatial context, here we are detecting the mRNA molecules directly in the tissue. So that's going to be enabling single cell or singular cell resolution with this. So stay tuned for those newcomings, but let's focus back on the Chromium single cell workflow or the solution and what it comprises. So this is an end-to-end workflow brought to you to generate and analyze the single cell and nuclei data. It's ready to use reagents. The workflow is fairly robust. We have a lot of supported protocols, and it also comes with turnkey analysis tools that are free, which I think is quite important for most beginners. That analysis side of things is often considered very complicated and difficult as a starting point. And we have both tools available, the analysis, cell ranger or space ranger for the spatial technology. And we have a loop browser platform that runs off your laptop that allows you to interact with your data. But on top of it, you're not limited to that. So you can use any community available tools out there with the data sets that you generate as well. So when we look at the core of this Chromium single cell technology, we can see here in this image is this microfluidic partitioning that we are doing, and that we are generating these nanoliter droplets inside these microfluidic chips here on the instrument. And these droplets contain your single cell or nuclei. They contain a dedicated special 10x barcode and associated reagents. This occurs on the chip in an 18-minute run, so occurs super fast. And I just wanted to highlight here some of the key specs I think that I just want you to take away from today's meeting is you can run up to 8 samples on a chip. So you have 8 ports here, and you can actually capture a wide dynamic range of number of cells, and it goes up to 10,000 per well, so a total of 80,000 on an individual chip in one run. You know, we have a fairly low multiplet rate and quite a high capture rate as well. Something I want to mention to you today as well is that we've just launched our Chromium X instrument with the associated high throughput instrument. Now that instrument runs all the assays and kits that I just mentioned to you, also the 8-channel chip, but it comes compatible with a 16-channel chip. So that boosts your throughput, if you wish, dramatically, not only in the sample numbers, but also if you wanted to capture more cells. So here, this allows you to capture up to 320,000 single cells or nuclei per run. If you don't multiplex, and if you do multiplex, which we have cellplex available, you can actually boost this to up to 760,000 individual cells with this kit. You can switch between these, so you're not bound to running either or. But this is exciting new capability enabling, this instrument is enabled for all these technologies and assays that we cover today as well. Okay, so let's talk about single cell VPrime gene expression. Here, if we are looking at this kit, at the core of this is that we capture or the analyte that we are capturing here is your mRNA transcript, polyadenylated mRNA molecules we are detecting with this kit. Excuse me. But that's sort of your basic starting point. This 3' gene expression solution enables you to add in additional analytes by utilizing something that we introduced and called feature barcoding. But what that basically means is that on top of your mRNA transcript capturing that you're doing of your cell, you can now also capture the cell surface proteins. So you can detect cell surface proteins. And how that works is you're utilizing specific antibodies for your feature protein of interest, and that is going to be that has to be conjugated with an oligo, so that it can be detected by our assay chemistry. So you're really, in theory, not limited by any number of antibodies that you can detect or cell surface proteins that you can detect. We typically see, up to 100 or a bit over 100 antibody mixes being used with this technology. We partnered with BioLegend, and their Total-seq-B product portfolio is actually already directly compatible with our instrument, with our solution, as it has the correct oligo already conjugated. They are pre-titrated and ready to use with very little optimization. If you utilize that very same feature barcoding technology, you can add another type of analyte to this gene expression assay, and that is detecting CRISPR perturbations in the cell. How that works is that we are here now introducing a small oligo sequence into the actual guide itself. It can be read by our biochemistry of the specific assay, and it allows you to not only identify the specific guide itself of the individual cell where it's expressed, but because you are capturing the transcriptome at the same time, you can assess the actual perturbations that occurred based on the presence of that particular CRISPR guide within the cell. So really comprehensive types of data you can generate with the single cell VPrime gene expression kit. There is a large number of publications out there showcasing several of these combinations of the feature barcoding capabilities. This is just one here we picked to showcase how it can be used in cancer, specifically to deeply characterize the lung tumor microenvironment in tissue sections. So again, we have no time to look into the nitty-gritty of these publications, but it's there. And again, in our dedicated seminar, we can dive down deeper into this. So next up, let's look into the single cell immune profiling solution. Here, instead of looking at the 3' transcripts that we are doing in the single cell gene expression solution we just talked about, we are now capturing the 5' end of the mRNA transcripts. What that allows us to do is now, in addition, actually capture and generate the full length and paired VDJ sequences from T cell receptor and B cell receptors. And how is that achieved? We do this by a specific targeted enrichment of these sequences in order to pull this out of your individual sample. And again, also this technology is fitted out with a feature barcoding capability, allowing you to detect cell surface protein expression. Similar principle to the 3' gene expression, that oligo that is attached to these antibodies compatible with the solution are slightly different. So BioLegend, again, our partner with this specific capability, has developed the Total-seq series of antibody products that are compatible with the 5' immune profiling solution, allowing you to detect cell surface proteins on your cells. And in addition, also very exciting is to be able to detect antigen specificity by utilizing antigens that are also labeled with these compatible oligos, and that's, for example, provided already compatible to our solution by EUDEX. Just very briefly telling you what does that mean, what does it achieve if I add all these layers on top of my gene expression. And that's showcased here with our PBMC data, where on the left, the t-SNE or UMAP plot that you see where cell clusters are labeled in different colors, but also annotated just simply by utilizing gene expression information. is specifically when you look at the center cluster, fairly, it's not very specific. So while there is different cluster colors being pulled out, it is still sort of localized all in one place. But what you can see is when you add the cell surface protein information to this data set and perform the annotation that way, you can see that you can actually improve the annotation dramatically and make it a lot more clear in your data set. And what the T cell receptor and B cell receptor sequences look like, because we capture the full length, you don't have to do any computational calculations. We can actually detect, or we get the sequence and we can detect the phenotypes. And we can also look at the full length IG sequence for the B cell compartment, and also look at the compositions of light and heavy chain there. And an example of where this kit is utilized, specifically, I picked this one because it highlights, it's done with a B cell receptor and antigen mapping on HIV, and it is done utilizing a technique that some of you may know as LibraSeq, linking B cell receptor to antigen specificity. Again, this technique is slightly adjusted and designed to look at B cell receptors and their antigens. And I've picked this example because 10x is going to bring out virtual assay, basically utilizing this actual workflow, which we call BIP. So that's coming next year. Okay, now switching gear from 3 and 5' gene expression, capturing whole transcriptome, to the capability that 10x developed to also perform targeted gene expression. So if you're only interested in looking at a specific cancer type, or as in immunology, a specific innate and adaptive immunity components, we have developed these pre-designed panels that you see here for these sort of 4 key areas, containing a large number of genes. So on average, over 1,000 genes each focusing on very specific key points within these research areas. You can see actually a detailed list of each individual gene captured in each panel on the website. If you follow the link, or please email me if you need the link. But what you can also do, the enabler, is if you take these panels and you're not quite satisfied with their content, you can create an account online. It's very simple, and utilize our custom panel designer tool. You can adapt these panels and add up to 200 genes to each, or you can create completely custom panels from starting a minimum of 10 beta 50 genes up to 1,500 of your own. The steps are fairly straightforward, and you're left with a list that you can submit to order straight away. And you can also add in your exogenous sequences if you need to. So targeted sequencing to focus on the genes that matter, at the same time as also saving cost on your experiment, is enabled with our technology. So the last product, or the last assay I want to mention to you today, is our epigenetic solution, our combined attack or assay for transposase accessible chromatin plus gene expression kit, and we call this kit the multi-ohm kit. So with this kit, you are able to profile the transcriptome as well as the chromatin accessible structures within cells. So we are using cell nuclei as input for this particular assay. So what this allows you to do is, you can look at transcription factors, and you can identify a whole range of mechanisms that underlie specific activation paths, for example, in a disease model, if you're looking at disease states. What this allows you to do is by capturing these modalities together. So some of you might be aware you can do gene expressions separately, and you can perform an attack-seek experiment separately, and then you have to perform linkage analysis or invert linkage analysis on this. This kit allows you to do both gene expression and attack-seek information from each individual nuclei used, and then from each of them you get these linkage relationships between the chromatin structures. So here, this is just a brief snapshot of what Loop Browser allows you to see with the multi-ohm kit here, focusing specifically on one gene at the top, ACLEF1, and some of the linkages that are positive and some of the linkages that are negative are highlighted here in the different colors. And then you can look at the specific peaks that are captured in the specific cell types of these accessible chromatins. So again, this is something that we can not cover in much more detail how it works and the intricacies of the data you get, but again, we dedicate a seminar to this. So to finish this up, there is a number of additional products coming as well to keep advancing what we offer and to keep making this technology even more accessible with the fixed RNA profiling kit, as I mentioned to you. But again, I wanted to highlight that if all of this was a bit too still too confusing, please don't hesitate to contact me, to contact any of us here in Australia. There is a big team available to support and answer any additional questions. And with that, thank you very much, and I look forward to hearing Luciano's presentation.
Well, thank you very much, Yvonne. That was great. I think that gives us exactly what we wanted to be at this point, with a big overview of what are the possibilities, and very exciting that pipeline that you had at the end with all the new products coming up. So thank you very much for your presentation. Now we have Luciano Martellotto. So Luciano, he's actually in Boston at the moment. So he is the director of the single cell core, the Harvard Medical School, and he's coming today to talk about a little bit of his experience on handling many different samples. And Luciano has been involved with single cell genomics pretty much since the beginning of the technology. He was based in Melbourne, then he moved to Harvard, and then I can also announce that he's coming back to Australia. So he's joining the South Australia Immunogenomics Cancer Centre, which is part of the University of Adelaide. And he's basically joining forces with Jose Polo, which is the director of the Adelaide Centre for Epigenetics. And he's setting up the lab next year. So he's looking for recruiting both wet lab experienced people and also bioinformaticians. So anyway, it's a pleasure for me to introduce Luciano, and over to you, Luciano. Thank you very much for being here.
Thank you very much, guys. It's, as you always, it's a pleasure to talk in these kind of seminars, and I'm very excited to present what I have to say today. So the presentation of today is going to be a little bit different to what you will probably normally hear. I'm going to talk about how to do an experiment, but I will make you aware who are your best friends in a single cell when you are planning a single cell experiment. So for those that don't know me, my name is Luciano Martellotto, and I'll hopefully I get to interact with you in the future. So I'm going to start just by letting you know that, let me see, there you go. So when you try to plan an experiment, one of the things that you need to understand is that planning an experiment for a single cell is not yet trivial. A lot of people think that is, you just put this in the Chromium machine and that little thing does everything. It is true that it does a huge amount of things, which it is encapsulated and individualized in the cells, but there is a lot of things that you need to take into account in the context of single cell experiments in general. So the single cell experiment starts way before running the samples in the Chromium. So there is a huge amount of steps that can go wrong. And for these experiments to be successful, every single one of those need to work properly, okay? And so, and to me, you know, coming to Harvard to work and to help the core, because the core, I will explain how our core works, but he's been kind of enlightening because I was exposed to a lot of samples before, but the amount of different type of materials that I've seen here and the huge amount of variability across all researchers and samples kind of opened my mind and realized that if you want your experiment to succeed, you need to find your right partner. Okay, so the right partner to work with you. That is on the web side of things and also on the bioinformatics side of things. And I will make sure that that is clear at the end of the talk. So, and what I want to put, the message that I would like you to see at the end, or to take home at the end of this, is that single cell core facilities or platform technologies like the one that I am actually directing here, or the one that David is directing in Sydney, are exceptionally well positioned to actually helping you for your experiments to succeed. And the reason for that is because we are experiencing across a multitude of samples, different type of technologies. We get to see different type of researchers and projects, and the way that people interact with those projects, and that give us a little bit of a breadth in the understanding of the experiment itself. So I think if you're really looking for a partner, core facilities nowadays, especially single cell core facilities, are specialized on making sure that you have a successful experiment. So I'm going to talk a little bit of you know, what's the workflow that we have set up for the single cell core laboratory here at Harvard Medical School, and to the point that it's very successful. It's a very successful core and very busy, and I think it's probably one of the most rewarding compliments that we have heard from our collaborators or users is that they felt that we were an extension of their own lab, which when you are trying to help people to design their own experiments, that's probably, it doesn't get better than that compliment. So the single cell core has two parts. One is the routine work, which we have a team of 3 people that is continuously doing, you know, running the samples for different platforms that we support. And there is an R&D team, which is the one that I basically handle, which is essentially implementing new technologies. You know, whenever 10x come with a new kit, or we get to do beta testing, or there is a new platform that we want to test, or there is a project that came from a particular researcher that want to develop, but they don't have the understanding of how things work, we help them get there. And then in the last year, we actually run 170 projects, new projects, right? So we usually have a recurrent project of people that keep running projects, but there were 170 new projects from 60 labs. So that is for 4 people. That's an immense amount of work. And this year we have already, completely passed that number. And even though COVID hit, people kept doing experiments and spending a lot of money. I understand the difference between Australia and America. There is much more money here. People just go crazy and run a lot of experiments. And one of my jobs is to be the goalkeeper then and telling them when to do an experiment, when not. But that doesn't stop them to do a lot of experiments. And that's what we have 170 projects in last year. So we have collaborators in the US and international. And we also have, in terms of tech dev, we have collaboration with different groups, also national or international. And most of these are projects initiated by the collaborators or with other companies that we have a sort of relationship as well, and get these projects going. Essentially, this is the part that I mostly involved. So these are the technologies that we have available at the moment. So obviously we have 10x technology. This is the one, I would say, probably the battle horse for us. When I moved 2 years ago to Boston, the main technology was the in-drop system, which, for those that don't know, this is sort of like the previous version of, or one of the first version of microfluidics. But then today we have, I think, probably 95% of all the projects that we run are 10x, and we do every single application from 10x, and including Visium, spatial transcriptomics. And we also run a few beta testing as well. And then we also have the VD Rhapsody. What we haven't, we have implemented it. It's very successful with a few particular customers, but again, it doesn't not have yet, and I don't think it's going to have the access that the 10x technology have, because as you know, it's very versatile. You have a huge battery of technologies or assays that you can potentially run. So the goal of our lab, obviously, we want to make sure that your experiment is successful. All right, we want to make sure that your experiments are successful. But this is just not only on us. This is something that we have to collaborate. So as a core, we make sure that we collaborate, that we have things clear. So we define that, and for that, we have developed this kind of bulletproof workflow that I hope I can portray that for you. So this is how all the how our workflow works. If you want to see, I have a very nice animation here made by my friend Katia Moutinho, that it actually goes through all of these parts and explains every little part. But I'm gonna go into step playing this, because otherwise I will have nothing to say at the end. So the first part, I think it's probably the most important part, is that, especially for us, for those that actually want to understand what your project is about, we have a brief questionnaire. So there is an application online that people can access, and there is a series of questions. It's not a very long questionnaire, but it does have information that allows us to understand more or less what type of experiments you want to do, what technologies you want to use, what type of samples, and especially if you do have experience or not with single cell, because then we plan the second part, which is the consultation. So we do have a consultation, which is a 1-to-1. Sometimes it's usually 2-to-1. But one of the main things that I insist, we need to have a series of key players. All right. So it's very rare that we have two of us and one of them, because I try not to have any consultation or not schedule a consultation unless there is a bioinformatician person in the meeting. We do put a lot of pressure on that. We make sure that the bioinformatics person is there, because it's the person that is, at the end of the day, is going to do the analysis. So we want that person to be present. And then the PI. The PI is optional, but ideally we want the PI to be present, because he's the one that puts the money. So, and he's the one that at the end of the day will decide how many replicates you're going to end up doing. Okay, it doesn't matter how many times I tell them you need to do replicates. They are the one that decide how many they're going to end up doing. And in this consultation, we essentially go through. We ask them to present us the project, to justify the need of single cell. You know, there are a lot of projects that do not need single cells, and for those projects, we actually advise them not to run. And it doesn't matter whether they want to run it or not. If there are some projects that are more or less single celly, but if they are not by any means single cell, we just do not run it, okay? But in general, we do support 99% of all the projects. So we discuss everything from the type of samples, of how the samples were dissociated, how are you gonna dissociate it, what protocols are you gonna use. We discuss whether you want to run replicates, how many replicates you do. Are you going to use CellPlex? Are you going to use 3', 5', VDJ, site-seq, multi-seq? We discuss all of that during this. We do advise what's the best platform for that particular project. There are platforms in which there are particular, very specific projects in which 10x is not applicable. Therefore, the 99% of the rest it is. But it is our job to be able to give them the right advice. And of course, we discuss sequencing and costing, okay? But this is the part in which we understand how a project works and we help them design the project. It doesn't really finish in this first meeting. We usually get, 2 or 3 emails back and forth because we need to make sure that they got the message. This usually is an information overflow, but we definitely give them a lot of information to take home and digest. Then the next part is my favorite part, which is the part in which we discuss the sample preparation. Okay, this is something that happened post consultation, and in this part we usually recommend. This is the part in which they run a pilot. For the majority of our projects, we do recommend to run a pilot, and a pilot doesn't mean to run an experiment that is identical to your final experiment. It means that you have to run a representative experiment that will give you information to understand whether your sample is of good quality, whether the number of cells that you profile is sufficient or you overkilled it, to determine whether the sequencing that you are planning for your particular experiment is enough. And whether you time the experiment right, because some people may want to do some enrichment and they wake up 3 in the morning, they kill the mice at 5 p.m. and then they bring the samples at 3 p.m. in the afternoon, and that it's a very long experiment. And I can tell you, I can see them in their face, they are completely knackered by the time they get to the lab. And this they suffer, but the samples suffer more than them. So what we discuss in all this process is how to make sure that they understand the timing of the experiment. Okay, so they need to be able to coordinate course of facilities and understand how the whole process works. And after they have that particular experience, they're going to take all these results and they are going to adjust the following experiment. Sometimes may require another pilot. It's very rare, but sometimes it happens. And then after that, you just go and you do your final experiment. But we do have that pilot to be able to adjust all the things that go from timing to the number of cells that you want and the type of the dissociation protocol to make sure that the final experiment is successful. All these 11 things that I just enumerated here, for example, these are things that we usually discuss with this. So we want to make sure that they understand that all the little points that are important for success. And this is important for a particular thing. I'm gonna do something that I'm gonna cite myself here twice just for you. And this is one thing that I always tell them. 80% of your experiment, it doesn't matter what type of experiment is in the single cell or on the spatial transcriptomics space, depends on your sample prep. If you do have your sample prep nailed down, there is a huge, I say 80% because I want to be nice, but I would say 90% of your experiment will be successful, because that's the part. A good sample always will help you. If you don't muck up at the end, is going to give you good data. Okay, it may not be perfect the first time, but everything becomes easier with practice. And the other thing is, you are going to find a lot of protocols available online. 10x has a range of protocols, demonstrated protocols, and also user demonstrated protocols that they made available. There is a lot of protocols that we have made available for everyone. And I will show you there are some protocols from protocols.io that you can access, or the Single Cell Ninja, which I tell you what that is as well, that are what we say optimized. Optimized means that we have found the right time, the right speed or the right G's of your centrifuge for you to run it and to run it successfully. But it's not going to be run successfully if you, the user, is not optimized for the protocol. So it's a combination of both things. So that's why the pilots are so important, to be able for you to practice the protocol, because things are always, like everything that you do for the first time, is always a little clunky. It could be nice, but it could be clunky. Okay. And as I said before, we have made a lot of protocols available. This is some of the protocols that I made available for nuclear prep, for example, from the very beginning, and that that I started with single cell, and now we Katia Moutinho, we have sort of implemented this platform called the Single Cell Ninja. This is a free platform in which you get access to seminars, you get access to all the protocols curated by us and by some other helpers, in which you can access to protocols about single cell or spatial transcriptomics that we think you should know. Okay. And of course, protocols.io is a beautiful collection of a lot of protocols. Everything is in there. So we do give advices of also during the consultation or post-consultation, what is the best protocol for their sample, if available. Now the next part it should be very simple, okay, which is the booking of the single cell run or the spatial transcriptomic run, and that should be the simplest part. But unfortunately, in our case, and this is real story, 80% of our users do not get to the lab on time, which makes things extremely difficult for the core facilities, because we run on a tight schedule. We have 3-4 collaborators wanted to run their experiments, and that becomes really complicated. And especially in a COVID time in which we had to run every single sample. Now we train them and they run them by themselves. So we just sort of like overlook, but it's very important that, a single cell core facility is as important as a fax sorting facility. Okay, so in our experience has been a real struggle. Now then we plan the single cell experiments. As I mentioned before, we have what we call staff assisted, which essentially we do everything from them. If it is single nuclear RNA-seq, I prepare all the single nuclear RNA-seq that go through the lab. But if it is single cell, sometimes we don't. Sometimes we let them prepare. But essentially, if we can prepare the samples, if we know the sample, yes. Now, if someone comes with vein from zebrafish, I may not be able to know how to dissociate that, and that's where the pilots are important. Okay, but otherwise, we take care of pretty much everything in the lab. And then the other one, there is a self-assisted one in which we train them and they just go ahead and run it by themselves. We usually, this is great for us, and especially is because before 10x, or at least until 10x come up with the RNA-fix solution, most of the researchers especially now we are affiliated with Dana-Farber, which is a cancer center here. So we get a lot of tumor material that come from the operating theater, right, fresh. But the surgery may happen at 5 p.m., 7 p.m., 10 p.m., midnight. And to be honest, I cannot do that all the time. I can do it one-off, but that's it. So we are really looking forward to the RNA-fix solution, because it's going to help those guys tremendously. Because at the moment, what they have to do is just to fix this, to store the cells, to cryopreserve, that it will, unfortunately, will render a lot of dead cell death, or they have to do single nuclear RNA-seq, which is great, it's fine, it's very useful. But if they can do whole transcriptome from cells, that would be even better. So I particularly looking forward to the RNA-fix part. Then the next part is the elaborate preparation. So up to 3 weeks is more or less what that's the time. We usually have it ready by 1 week or so. We try to pull a few libraries together and making sure that none of them have batch effects, because if you make libraries from the same thing differently, you will see the batch effect 100%. So we try to put things together from one particular user to make sure that there is to reduce the amount of batch effect in those cases. But we do finish the libraries in 1 or 2 weeks. But we realize that the success, the secret of happiness is setting the expectation right. So every time that we tell them it's going to be 2 weeks, on the day 13th, we get an email say, "Are my libraries ready?" So we tell them 3 weeks. We know they're going to finish earlier. So if David tell you that it's 2 weeks, you can be sure that he's going to finish in 1 week. Sorry, David. So the other thing is, there's a few other things that we include here. We do a lot of QCs. If we do the sample prep, we take photos at every step, or we do bioanalysis of all the libraries or type stations across the entire workflow. And also we do, while we're not a sequencing core, we do have collaboration with 4 or 5 cores around in the Longwood area, which is what the Harvard Medical School is, and we do offer this pooling and and submission for them, and then we get the data and then we send the data to them, and we do the multiplexing, and now in the case for their case, they can choose whatever sequencing core facility that they want. We don't really care which one they do. We do have some preferences, but we don't really tell them. And in some of these cases, we usually do recommendation of how much they need to sequence. We do all the calculations for them and tell you, okay, this is how much you need to sequence, because we are the ones that really know. I would say 90% of all the users that I've interacted, they don't really know exactly how, how much to sequence. And then in terms of the analysis, as I mentioned before, ideally they will have a bioinformatician lined up. We also have a partnership with the Chan Bioinformatics core. These guys are extremely busy, and which pretty much every bioinformatics person in the world. So usually, they struggle a little bit with that. But lucky for America, and I'm hoping that this is going to be happening soon in the in Australia. In America, we have the 10x cloud, which essentially it allows you to upload your FASTQ on a cloud, right? And then you can perform the analysis of hundreds of thousands of cells in a matter of hours. It's insane. I cannot explain how good that is. I think I've said it many times in other seminars that, I had an experiment of 50,000 cells and I got the analysis ready to check how, the web summary or the loop browser files in 45 minutes. 45 minutes, that it's insane. That is not something that, that would normally happen in a normal class from any place, standard place, like from a university or from our core facility. So 10x Cloud is something that I recommend once it's available in Australia. So, and I said, this is essentially how our core looks like. But the part that I've been implementing lately is essentially I get the data, okay, from the sequencer. I upload it into the cloud because you can do that. You don't need to know code, so everything's explained exactly how to do it on the website. You need to use the terminal, but it's very simple to do. At first, it was a little bit. If you know how to use Unix or R, you will be able to do it. I struggle at first, but then it's super easy. And then what I do is I take the data, I look at the web summaries, and I don't get into the gene expression or anything with the browser or anything like that, but I do have a look at the quality because I want to be the first person to tell them your experiment was great or your experiment is a disaster. And I want to be able to have some sort of argument, because what happens is that you get to that point and you don't hear from them for 3 months, and then they come back to you and say, my data is really bad. And sorry, who are you? What experiment are you talking about? We just ran 200 experiments in the last month. So I prefer to have that information up front, and it's been very, very nice to have that discussion, because I've learned a lot and they learned a lot as well, to understand all the metrics that you see on a web summary, which I think if you get to understand that, the day that you understand a web summary, you understand how the 10x data works, and that's beautiful. Essentially, just to summarize, for your experiment to be successful, my suggestion is just to partner with people that actually are knowledgeable. It could be a research lab, that have time and they want to help you. But I strongly recommend you to go to core facilities like the one from David, that will essentially help you design the experiment from the beginning to the end. That will help you understand what data you are seeing, okay? And it's a commitment from both. It's from you and from the core, okay? There is part that you have to do and part that the core have to do. And if you do it that way, your single cell experiment is gonna be a nice experience rather than a nightmare, all right? And with that, thank you very much for listening to me.
Well, thank you very much, Luciano. That was great and really clearly got a bit of an overview of how a core facility works, what people can access, and especially what is the interaction user and expert in the area. So I will also add to the comment from Yvonne earlier that you also have access to 10x support for that design of the experiment. So, if you're interested in doing this, these experiments are expensive. They're very rewarding. So you want to design them very well and get the most out of it. So I was just a little bit scared, Luciano, when you mentioned the key players on an experiment. I thought that you were starting to talk about Messi and, Real Madrid and all those things. And I was like, he's Argentinian. We're going to get in trouble here. Sorry.
Yeah, well, no. I wanted to believe that I was the Messi, all the time.
So, look, I think, so I would like to thank you both for the presentation and for your time. This is fantastic. And I would like to open for questions. So there is a poll coming out. I also got in the chat, as in case that you didn't see, the 10x Cloud, Yvonne just announced that it's coming to Australia soon. So I will have access to that service as well. So that's fantastic. And yeah, so I don't know. I mean, is there any question that you want to ask? Please, is the moment to do it. And if like in the meantime, while we're getting questions from the audience, I would actually like to. I was really interested, Lou, on your comment on the sequencing, like where you have, like you do all the preparations, like people kind of sometimes is not clear on, because when you say single cell sequencing, and people think that the sequencing actually happens at the same time. Yes. And it's a secondary process after you do all the barcoding and cell partitioning and the molecular amplification and library prep. So this is a very similar setup. We don't have at UTS a very strong sequencing capacity. So we outsource our sequencing, and this is the plan to do. And I think that is a very good point for people to clarify that we don't sequence. We actually prepare the samples to be ready for sequencing, which is a bit different. So that's a very key point, and I'm very happy that in Harvard that you guys do the same. You don't really need to have.
We decided that having a sequencing instrument today, with the way of how the throughput on the single cell is changing, it's not possible, because we need to go from 2 years ago we would have started with 3 years ago with an NextSeq, and then we would have been short. We have to go to a NovaSeq, and then they said, you know what? No, it's easier for the user to go and access to whatever core they feel more comfortable. Some of them they have discounts, because it's part of affiliation to a particular university, and just go and get that discount. We don't really think that is necessary, but we do really emphasize that there is a huge difference, especially they totally forget, you will find it as well, that they totally forget that the sequencing also has to be paid. so they get enthusiastic with the number of cells. I want to do 50,000 cells, and I say, hey, you have to sequence 50,000 cells. That's right. So but yeah, so I think it's so we do discuss sequencing and sequencing cost significantly, and people is happy to pay for the premium cost of 10x because they know that it works. Okay, and if it doesn't work, you're gonna get the support that you need, but nobody wants to pay extra and sequencing. So that's why we do take that seriously.
You know, that's a very good point. I think I mean, it's logical from the point of view when you know the technology, but from the newcomers, sometimes it's a bit confusing on that. And another question is perhaps for Yvonne in terms of accessing. So who will be the best person to contact from 10X to start kicking the ball rolling in a design of experiment?
So maybe I just bring up the slide again. So it can be any of our science and technology advisors. So I can get the ball rolling with what Luciano mentioned, you know, like the initial design conversations. What do you want to achieve? What are you thinking? What sample types do you have, etc. And then I would bring in either Catherine King, who is our field application specialist, based in Melbourne, or Paul Gooding. He is the field application specialist for Millennium Science. So he's in Adelaide. So depending on, you know, who's available, we have these two experts. And we usually do these meetings jointly because the FAS can bring in a lot of the expertise on the actual handling, you know, all the timings that Luciano mentioned, etc. we can discuss. So I think I'll bring that up again, but if they get in touch with me first, and then I'll sort of invite the appropriate people along.
And just to follow up on that, and what Yvonne has presented here in terms of, you know, the 10X portfolio of different kits and different I guess questions that you can answer or you can ask to your system, and also from Luciano's point of view, I just wanted to just clarify to everybody that we have that technology available and is ready at UTS. So this is nothing. This is not on development. This is actually something that we have already established and implemented here. So it's ready to go. We are ready to go if you are ready to go. So I'm not sure if we have any more questions in the chat. I don't think we do. So with that, I would really like to thank you both, Yvonne and Luciano, for taking the time on presenting today. And Lou, I know it's late over there, so thank you. Really appreciate that you're taking that time. And yeah, I'm looking forward to the next one. So we will have another seminar in 2 weeks' time. So just watch the space. Thank you very much, everybody.
Perfect.
Thank you.
Thanks, David.
Thank you, guys. See you.
Thanks, Luciano.
See you guys.
Bye.
No worries. Thank you.